Assalamualaikum and hello everyone. My name is Muhammad Irfan bin Muhammad Zabidi and my MATRIC number is BC252542. So today I will explain about the special sense organ. So this is my table of content for this video assignment, which we will focus mainly into the anatomy of the special sense organ. First of all, special sense utilize specialized receptors housed within complex organs located exclusively in the head. This completely distinguishes them from general somatic senses like touch or temperature, which are spread throughout the rest of the body. These senses are vital for three primary reasons. First, they provide environmental awareness by helping us detect light, sound, chemicals, and spatial orientation. Second, they ensure survival and protection by helping us identify hazards and navigate safely. Finally, they enhance our quality of life by facilitating human communication, stimulating appetite through taste and smell, and triggering memory cells. Now let's look at the anatomy of tongue. The tongue surface features four types of papillae. First, filiform papillae are distributed across the anterior two-thirds of the tongue. They lack taste buds and serve a mechanical role. Second, fungiform papillae are mushroom-like projections located on the tip and sides, containing a few taste buds each. Third, velate papillae are large structures arranged in an inverted V at the back, housing the majority of our taste buds. Finally, folate papillae form weak ridges on the lateral sides, losing their taste buds after early childhood. So, when we eat, chemicals dissolve in saliva and enter a small opening called taste pore. This activates the gustatory macrovilli on the receptor cells, which send sensory signals through three cranial nerves: the facial nerve for the front two-thirds, the glossopharyngeal nerve for the back third, and the vagus nerve for the pharynx. This pathway allows the brain to perceive five basic taste sensations map-coded across regions of the tongue and throat, which is sweet, sour, salty, bitter, and umami. Next, we move into the olfactory organ. The primary anatomical site for the smell is olfactory epithelium, found at the very top of the nasal cavity, just beneath the cerebriform plate of the ethmoid bone. Within this epithelium, we find three critical cell types, which is the olfactory nerve cells, which act as receptor, supporting cells that protect and maintain these receptors, and basal cells, which is function as stem cells to replace neurons continuously throughout our life. So how it works? When we inhale, airborne chemical molecules dissolve into the nasal mucus layer and bind to receptor proteins on the olfactory heads. This triggers a nerve impulse that travels up through the tiny holes of the cerebriform plate via cranial nerve 1 and the olfactory nerve. The signal reaches the olfactory bulb and travels down the olfactory tract to the brain, directly targeting the olfactory cortex in the temporal lobe for conscious perceptions, as well as the limbic system for emotional and memory responses. Turning into the eye, it is structurally organized into three concentric layers and three major chambers. The outermost layer is the fibrous coat, made up of tough, white sclera and the clear, avascular cornea that allows light to pass through. Below this lies the vascular coat or uveal tract, consisting of the blood-rich choroid, the muscular ciliary body, and the colored iris, which dynamically alters pupil diameter to regulate light entry. The innermost layer is the retina, which is houses of our sensory photoreceptors. Internally, the eyeball is split into fluid-filled spaces. The anterior and porous chambers contain clear aqueous humor to nourish and protect the eye, while the massive vitreous chamber is filled with jelly-like vitreous humor that maintains the eye's shape and structural integrity. The auditory system processes our senses of hearing using three distinct regions. First, the outer ear uses the pinna to capture sound waves, funneling them down the external acoustic meatus to vibrate the tympanic membrane. These vibrations pass into the middle ear cavity, where the meatus inserts and staples ossicles physically amplify the energy and push it through the oval window. These movements set fluid waves in motion within the channels of the inner ear's snail-shaped cochlea. Inside the cochlea duct sits the organ of Chaudhari, our true receptor organ for hearing. As fluid waves causes the basilar membrane to vibrate. It bends the specialized sensory hair cells, trigger nerve impulses that travel along the vestibular-cochlear nerve directly to auditory cortex of the brain. The ear's role in equilibrium and balance is managed by the vestibular apparatus in the inner ear. The system is anatomically split to track different types of movement: static equilibrium, which measures linear acceleration, and gravity, like tilting your head, occurs within the vestibules utricle and sicule. Here, heavy calcium crystals called otoliths slide across a gelatinous membrane, bending hair cells to signal positional changes. Dynamic equilibrium, which tracks head rotation, is monitored by the three semicircular canals. When your head rotates, fluid inside these canals pushes against the gelatinous cap called cupula, which sits inside the specialized swelling cells, the ampulla. Bending this cupula stimulates underlying hair cells, sending immediate spatial orientation signals down the vestibular nerve of cranial nerve 8 to help body maintain its balance. Now, let's review common sensory disorders. In clinical practice, we frequently encounter disorders across all four spatial senses. For vision, this includes myopia or nearsightedness, where light focuses in front of the retina rather than directly on it. We also see anosmia, the loss of smell, and ageusia, the loss of taste, which are often closely linked due to shared neurological pathway. As a nurse, our implications are critical. We must accurately assess sensory functions using tools like otoscopic exams and targeted cranial nerve testing. We are responsible for patient education, helping individuals adapt to permanent sensory loss safely, such as ensuring a patient with anosmia has functioning home smoke detectors. We must also advocate for prevention, teaching patients to use protective eyewear and avoid environment-induced loud noise exposure. So in conclusion, our spatial senses are irreplaceable: pathway for survival, danger detection, and human connection. Through early nursing intervention, through assessment and robust patient education, we play a definitive role in preserving these vital functions and maximizing our patients' overall quality of life. So that's all. Thank you for your time.